Nice model because the renal corpuscle, so this is the most detailed we're going to get with the nephron, and it really is kind of maximum detail. If there's any more detail than this, you're going into the cell biology of it. So I want to make sure you guys are clear on the difference between a renal corpuscle, which is what the title of this model is, and what a Bowman's capsule versus a glomerulus is. Because a lot of times we just throw around these terms as if they kind of mean the same thing. So let me explain. The most general one is renal corpuscle. So renal corpuscle is everything in this image except for down here. This is the proximal convoluted tubule. That's not part of the renal corpuscle. So ignore that. And then ignore number 11, which is the distal convoluted tubule. That's also not part of the renal corpuscle. But everything else is. So it's very general. The renal corpuscle is mostly made of the glomerulus, which is the capillary bed, and the Bowman's capsule, which surrounds that capillary bed. Now the Bowman's capsule, that's it for the glomerulus, but the Bowman's capsule has a couple other sub-layers to it. It's got a visceral and a parietal layer. So this Bowman's capsule, it's more accurately represented on this side. And so what the Bowman's capsule is like if my fist is the glomerulus, the Bowman's capsule is like a balloon that I just shove over my fist. So it's got a visceral layer that would be touching my hand, and it's got a parietal layer, which would be the outside part of the balloon that's not touching my hand. So it's this visceral and parietal relationship like we saw with the heart, where it is like a fist in a balloon. And so the part of the Bowman's capsule that's touching my hand, the layer is called the visceral layer of the Bowman's capsule. Illustrated right here, this kind of yellowy color here. That visceral layer is made of cells that are called podocytes. So if I ask you to name any of these cells, there's a nice nucleus right there and there. The cells are podocytes, but they make up the visceral layer of the Bowman's capsule. So I'll be as clear, I'll bold it and underline it if I want the layer versus the cell. And then the parietal layer of the Bowman's capsule is simple squamous epithelium here and here. And then there's the capsular, they have space on your sheet? Capsular space or?
Lumen.
Lumen, so you can call it whatever you want. It is an opening like a lumen. Capsular space, capsular lumen, you'll probably hear both terminology. But that's just the space in between. So that's it for kind of those subdivisions. Now the last thing I'm going to talk about in this image is what's going on at the top here. So you have what's called the juxtaglomerular apparatus, which is 13 in this image. And the juxtaglomerular apparatus is made up of two things. The macula densa, which are cells that are part of the distal convoluted tubule right here. They're just like these 7 cells right here are the macula densa. And then this right here, so this is the afferent arteriole coming in, and all arterioles coming into a capillary bed have smooth muscle there. It's what allows blood flow to enter or not enter the capillary bed. But this is like a really buff precapillary sphincter. But it has a special name, often referred to as the JG cells or the juxtaglomerular cells. They are extremely important, and when you talk about the renin-angiotensin system, this is the beginning of that sequence of events that happen. And so that's why these are important to point out. But these JG cells, juxtaglomerular cells, are just smooth muscle cells that have endocrine function. So there's a couple different ways that the kidney has endocrine function, but this is the most important way, in that when the blood pressure to the glomerulus all of a sudden drops off. Oh, are you guys done your renal questions yet? You're going to get one about renal stenosis.
Expands and contracts.
Well, it senses the pressure. It's a pressure sensor is what it is, the JG cells. So I think you guys are going to get a question in lecture. I don't think it's on an exam, so I think I can tell you this. I don't think it is. But it's about renal artery stenosis. So renal artery, that's the big artery way removed from this that's bringing blood to the kidney. If you have a narrowing of the big tube going to that littler tube, the pressure at the glomerulus will drop. And that's not a good thing because that is going to drop renal function. And so you need something in there to sense that pressure drop. And the thing that does that are the JG cells. When the pressure drops at the glomerulus, the JG cells feel, oh, there's less tension on me. And what they do is they release renin into circulation. Renin leads to, it's a couple different steps in there, but it leads to the recovery of water and the boosting of your blood pressure. Because when the blood pressure drops to the glomerulus, your glomerulus, through the JG cells, basically act to reverse that. And so the second class of drugs that is used to treat hypertension are called ACE inhibitors. They inhibit the renin-angiotensin pathway that begins right here. And so if you know someone that's on ACE inhibitors, they're blocking this JG cell's ability to raise your blood pressure, which can inhibit renal function, side effect, but benefits you in that you're going to have a lower average blood pressure over time. So together, just to review, juxtaglomerular apparatus, that's the combination of the macula densa and the JG cells. And in 2 weeks, we will talk about how this, and in lecture maybe next week, you'll talk about how these communicate with each other, but it's a little too much for today. But today, I think just focusing on appreciating what the JG cells are doing. They're really just a pressure sensor. And so I'm guessing your question in class, if you think about this, so someone with renal artery stenosis, which means narrowing, they're actually going to have a higher systemic blood pressure, but their kidneys aren't going to know about it. Because the pinching off of the renal artery will boost the blood pressure everywhere else in your body, but your kidneys won't know it's going on because they're actually getting lower blood pressure. So I think that's why he uses it as an example. So your kidneys won't be able to help you with your blood pressure issue. They'll actually cause you to have even higher blood pressure. It's not a good thing.
So the macula densa is only the DCT cells that are adjacent
right
to
there.
And
it's literally like 7 cells. If you look at it under a microscope, there are these darker cells. That's what the macula term means. It means dark, dense cells, macula densa. And so there's literally like 7 columnar cells abutted right up to the JG cells. And so don't worry too much about the function. I'm mostly giving you these kind of details so that you guys can make better use of lecture next time, because this is all really important stuff. So that's it for this model. You guys aren't doing any histology this week at all. So there is a section in the second to last objective, I think, that says no anatomical, structural, functional, and histological. Don't freak out. There's no lists on your sheet that are histological. The dissection we're not doing this week, but all of those terms I do want you to know. It's not going to be on a pig kidney. It's going to be either on a model or an image. And I think that's it. So let's